Hi, everybody. Uh, let's get started. The the things that we'll be discussing today are sort of from a time which I think was a very confused time in evolutionary biology. Uh, if we sort of go back to where we started from, we started with Darwin and Galton and their view of evolution. And up until that time, and then we had the debate between Mendelians and biometricians and the rediscovery of Mendel. And all this time, after discovery of Mendel, the concept of gene was a very abstract one. And as a result of that, evolution was primarily viewed as a morphological change. So when people were studying changes in characteristics of populations, they were interested in change in morphology and not too much more than that. And that quantified evolution. But then we had sort of the beginning of a time where theoretical population genetics and population biology took over, and people like Wright and Fisher came. And then we have these models of evolution, uh, concepts like fitness landscape. And evolution is viewed in terms of changes in allele frequency. And interestingly enough, the concept of allele is still an abstract one. We don't know the chemical basis of that. But it's a prism to view evolution in something other than a morphological context, while we still don't know what is the chemical basis of that context. And sort of 50s and 60s saw the transition from a view of morphological saw a transition from a morphological view of evolution to a molecular view of evolution. And now people were really starting to get interested to sort of go inside a cell and actually see what was going on. Remember genetic code, the concept of a codon and central dogma, none of that is known. All that is known at this point is that proteins are catalysts, and somehow this information is passed from one generation to another via DNA. So we know the molecular basis of genetics, molecular basis of genetics and heredity, and we know the actual chemical agent which causes these reactions. So the 50s and 60s, hence, is a transition, and it's marked by a lot of turbulence and a lot of ideas which were thrown out there. And we won't look at very much of it. We'll just spend today and Wednesday on this, and then we'll move ahead with it. But today is going to be relatively short. I would just introduce three different concepts to you. And then for Wednesday's class, I want you to come having read a paper that was published in 1969, a relatively straightforward paper, and we'll discuss that on Wednesday. And that sort of completes this part of the course. And then we'll move to more experimental ways to study evolution. But before I get started, any questions with anything that we have covered so far or anything in the course?
Is it fine if we ask questions related to the assignment?
Yes.
Okay. I had a doubt relating to SPT. So, in the first phase, when Wright says that due to genetic drift, you may move from a suboptimal peak to a valley, and this movement is a jump, right? It isn't continuous movement from the peak to the valley.
Wright doesn't say anything like that. I mean, it can't be too drastic a jump, because then its selection will be completely negated. What he means is that you enter into a phase where the relative effect of drift is stronger than selection, right? Uh, so he's not quantifying that how big a jump this is going to be. It's just that he's just saying that you move from a peak to a valley via the action of drift.
Okay. Okay. Thank you.
Yeah, sure. Others, anything? Traveling wave, anything at all? Okay. All right. So let me ask you this. Um, we were talking about clonal interference in a microbial population, and clonal interference meant that there are multiple beneficial mutations which are competing against each other at any given point in time in a population, and this competition between beneficial mutations has an impact on how fast or how slow evolution is going to go. Now it was sort of, and this is fairly recent. The paper, I mean, the material that we discussed was primarily from a paper that was published in 2008. So there is that. Another way to now we sort of go back in time, and we'll look at sort of how this problem was looked at in a diploid population, again in 50s. So imagine a population of diploid beings. So maybe let's imagine a human population. And if we look at a particular locus on the DNA, and all of us are diploid, so we are carrying two alleles at that locus on each of the chromosomes. The two alleles could be the same, or these could be different alleles. Now, suppose we were to sequence all of us in all of us at that particular locus. Do you think that we will find a more homozygous or a heterozygous populations? Or that's one. And second is, do you think we will find many alleles, or do you think we'll find one allele at a particular locus? And obviously, I'm speaking statistically. Uh, Suppose we were to do this exercise for 1,000 loci, uh, would we expect many alleles or one allele? And secondly, would we expect homozygous or heterozygous individuals? What do you guys think? Say anything that comes to mind. The question clear?
So there will be a distribution.
Okay, fine. So why should give me a reason for a locus where you will find only one allele and primarily homozygous individuals, and give me a reason for why you could find many alleles and heterozygous individuals. Yeah.
I think that if a trait physiologically it is related to a life or death situation, then the alleles would pretty much be the same. Most I think it wouldn't vary too much, but others like they don't really depend on immediate life or death situation. I think that gives survival or maybe other traits. Uh, I think they would have a higher variation
Okay.
because nature would allow that variation.
Right, right. What
Yeah.
you're saying is essentially that if the selection coefficient was too high, then you would expect a more homozygous
Yes.
distribution,
Yes.
compared to if it was low, then a more heterozygous.
Mm-hmm.
Fair, that's fair. What else? Can you think of a way we could experimentally characterize this? And think 50s, no sequencing.
If a trait is controlled by only one locus, then and if that trait is under strong stabilizing selection, then that wouldn't have many alleles. But if it's a quantitative trait like and many loci control that, there is more opportunity for more alleles to come up and be maintained.
Okay. Okay, sure. But how would you test this experimentally? So now what we want to test is, given for a particular locus, are there many alleles or just a single allele present in this population of 12 that we have in this meeting? Can we think of a way? Obviously, no sequencing allowed. Any ideas? Let's say we want to characterize presence of homozygosity, or presence of just same or different alleles at a locus which catalyzes a particular reaction that we know. So maybe glucose to glucose 6-phosphate. That's one reaction we know is carried by one enzyme. Suppose we want to test, whether we have one allele or many alleles at that particular locus. Any ideas? How many of you have run a DNA gel? One, two, three. Okay, a few of you. What happens in a DNA gel? How are you, or why are you able to visualize bands of DNA? Anyone?
Fluorescence marker.
What is that fluorescence marker?
Ethyl bromide.
Oh, not ethyl bromide, ethidium bromide.
Yeah, sorry.
Yeah,
Ethidium.
right. So ethidium bromide sits in the groove of DNA, and when you shine UV light on this gel, it fluoresces, and then you can make out which are the regions which contain DNA. All right, great. So I will discuss three ideas today, and then we'll sort of lay the ground for Wednesday, which is really where all the details we'll talk about them. How many of you have heard this statement that nothing in biology makes sense except in the light of evolution? No one has heard of it? Okay. All right. Who said that?
Is it Dobzhansky?
Dobzhansky, yeah, Theodosius Dobzhansky, who wrote a very famous book in the 50s. And Dobzhansky's argument is that nothing in biology makes sense except in light of evolution. At the 1955 Cold Spring Harbor lab meeting in the summer of 55, Dobzhansky presented a paper to the audience. And the premise that Dobzhansky is talking about, he's sort of laying down the problems in the field of evolution as we are moving forward. And remember, early 50s is the time when we're just coming out of the period where all this theoretical development has taken place, which and this theoretical development was preceded by a time where evolution was viewed as a morphological change that took place. And now, after this theory, there is a need for methods to be developed which allow us to characterize change in molecules inside the cell, and try and connect the changes that are taking place at a molecular level inside the cell with the morphological changes that are taking place at an organismal level. So Dobzhansky the paper that Dobzhansky read at this meeting was sort of I forget the exact title, but it was like on the lines of open problems in evolution and genetics that need to be addressed. And in that article, Dobzhansky presents what is called this is balance and classical hypothesis. In that article, Dobzhansky presents what are called balance and classical hypothesis. So this is lifted from his paper, where he says that at a risk of oversimplification, one may attempt to reduce the outstanding possibilities concerning the origin of the adaptive norm of two working hypotheses. And before he comes to this section, he's talking about what he means by an adaptive norm. For our purposes, we'll just think of adaptive norm as basically Dobzhansky's way of saying that of all the genotypic possibilities that exist in the population is what he's talking about, adaptive norm. And to further simplify it, we can think of just one particular locus. So the adaptive norm then is that what is this population comprises of at that particular locus. Does every individual have the same allele? Then we have every individual as a homozygous individual. Or do we have multiple alleles? And if we have multiple alleles, are individuals homozygous or heterozygous? There can be two cases where, suppose two alleles exist, there can be two cases where half the individuals are homozygous with one allele, half the individuals are homozygous with other alleles. Or the second possibility is that every individual is heterozygous with one copy of each of the two alleles. And of course, there could be intermediates between them. So all these possibilities is what Dobzhansky calls the adaptive norm. And we'll focus on the adaptive norm with respect to a population at a particular locus only. So Dobzhansky says that there are two possibilities of what could be going on. The first one, he says, he calls it the classical hypothesis. And in this view of evolution, what he says is that evolutionary change is essentially just replacement of one allele with another one. So when you're trying to connect this, you can try and connect this with the model of bacterial evolution that we talked about, and the picture where we saw that you have one beneficial mutation come up, and this beneficial mutation that then outcompetes the existing allele, and then you have a relatively long waiting time before the next beneficial mutation comes up. So in a classical view, what he says that evolution is essentially just replacement of one allele with a more beneficial allele. And of course, this would also include changes like that an allele that was working fine in the present environmental condition, you could suddenly have environmental changes take place, and this allele is no longer optimal for the new environment that has come over, and hence there might be a selection pressure for newer mutations to come up. So in classical view, the reason for heterozygosity is that newer mutations are coming up and the older alleles are being replaced. Hence, according to this view, Mendelian population is homozygous at most loci. We don't know what most is, but that's the premise that Dobzhansky is laying forward. And he says the reasons for heterozygosity in an individual are fourfolds. First one is, Dobzhansky says that sure, evolution is about one allele being replaced by a beneficial variant of that, but we know that deleterious mutations also take place, and these deleterious mutations will keep on taking place, and selection will keep on eliminating them. But in the time that selection takes to eliminate these deleterious alleles, you are going to have heterozygosity in the population, because if I as an individual carry a deleterious mutation, I'm going to be heterozygous. It's very unlikely that I'll have the deleterious mutation in both the alleles. So one reason for heterozygosity in the population is that I'm carrying a deleterious mutations in one of my alleles. And especially if that deleterious mutation is recessive in nature, then selection might take a relatively long time before that particular mutant allele is completely eliminated from the population. And that's pretty much it. Obviously, the rate of removal of a deleterious mutation is going to depend on its dominance relationship with the wild type allele. And I say wild type in quotes because who really knows what is wild type, right? So that's one reason for heterozygosity. The other reason for heterozygosity that Dobzhansky says is neutral variations. That I am a heterozygous individual, and one of the alleles is what is called the wild type allele. And again, wild type is something that is attributed to a allele if it's the majority allele in the population. So today, wild type might be something, but 1,000 years from now, the allele that might be carried at a particular locus may be completely different from what you and I are carrying, and hence that becomes the new wild type. So wild type is not a set in stone term. It changes with time. So I might be carrying one copy of the wild type allele and one copy of a mutant allele. But as far as fitness is concerned, or if you're talking of an enzyme, if you're talking of the characteristics of an enzyme, there is really no difference between the two alleles that I'm carrying. Right? So a characteristic of a protein enzyme are going to be its kcat, its Michaelis-Menten constant, and its stability. So if the two variants that I'm carrying are identical in these three characteristics, then the allelic variation that I am carrying as an individual is really neutral in nature. It doesn't come at a benefit or a cost for me to be carrying these two different alleles. The third reason that Dobzhansky says is called adaptive polymorphism. So you could have adaptive polymorphism if I am carrying two different alleles, and each of these two alleles, this could work in a couple of different ways. And polymorphism obviously means that I'm carrying different alleles, and this is adaptive in nature because it's possible that the two alleles, it could happen in carrying two different alleles can be beneficial in a couple of different ways. One is that I am carrying a particular allele which helps a certain physiological function at low temperature, and the other allele works very well at high temperature. So if I'm living in a place which sees very cold winters and warm summers, then for any season that's there in the environment, I am carrying an allele which basically works very well in the given environment. The other example where carrying two different alleles can be beneficial is the example of sickle cell anemia, that somehow carrying two different alleles makes you less resistant to a pathogen in this case. So this is what Dobzhansky calls an adaptive polymorphism, that just carrying these two different alleles is beneficial in nature, and hence you could see heterozygosity in the heterozygosity in an individual and multiple alleles being present in a population. And the last idea that Dobzhansky lays forward is the fact that the good mutations reaching fixation. He understands that there is a dynamics. I mean, the work of Wright and Fisher has made it very clear up until this point that once a beneficial mutation takes place, it's going to take its own time to reach fixation. And in this period of dynamics where one allele is being replaced by the other, you're going to have heterozygous individuals because this beneficial mutation is spreading through the population. Again, the dynamics is going to depend on the dominance relationship between the beneficial allele and the existing wild type allele. So these are the reasons of classical hypothesis, but it acknowledges that as a result of all of this, all of these four factors are a fairly minor player in terms of determining allelic heterozygosity. Right? Something like an adaptive polymorphism is going to be only relevant for a very select loci on the chromosome. For the most part, a Mendelian population is going to be homozygous in nature. That's the classical hypothesis. Dobzhansky doesn't say which one is true, he's just laying out the possibilities. At the other extreme is what is called a balance hypothesis. And in balance hypothesis, what you will see across a population is multiple alleles. What you will see at an individual level is two different alleles being carried. And this is going to be because polymorphism at an individual level confers some sort of an advantage at two different environments, and hence it's beneficial for an individual to be carrying two different alleles. And also there is neutral variation. So it's just sort of changing the relative emphasis that he places on the different factors which could be dictating the genetic makeup of a population. And if the adaptive polymorphism and neutral variation uh, components of heterozygosity and allelic variation are high, then you're going to have a situation where most individuals are heterozygous and there is considerable polymorphism in a population. And this hypothesis is called the balance hypothesis. What do you think of the two hypotheses? Which one resonates with you more, and for what reason? Are the two ideas clear? Yes, no,
The
don't
balance
care.
one, can you repeat the balance one?
Yeah, in balance hypothesis, essentially what you're saying is that there is going to be polymorphism at an individual level and also at a population level at a particular locus. And this polymorphism is driven because of neutral variation, that you have different alleles arising because of neutral variation, and also because there is adaptive polymorphism, that I as an individual benefit from carrying two different alleles at the at the same locus on the two chromosomes, and each of these alleles is going to be beneficial to me in one precise environmental context. So if I'm facing two different environments, then one of the alleles is going to provide me high fitness in one environment, and the other one is going to do the same in the other environment. And as a result of this widespread neutral variation and adaptive polymorphism, if I were to sample a locus at a population level, I am going to see many alleles. And if I were to sample this locus at an individual level, I'm going to see many individuals which are heterozygous in nature. So in the classical hypothesis, you're saying that at most loci, individuals are going to be homozygous, but that's not the case in the balance hypothesis, right? So what do you guys think? Anything?
The balance hypothesis sounds right.
Okay. All right. So, I mean, sure. So Dobzhansky doesn't know, but he says that there's a need for us to figure this out in order for us to understand how evolution is actually working. All these theoretical models are fine, but we don't really know the actual manifestations of what are the relative components of these two forces. So in order for us to find out, we need an experimental technique which allows us to quantify polymorphism at a locus directly, right? And that was sort of the need which was emphasized by Dobzhansky in this article. The next big leap came sort of almost 10 years after, exactly 10 years after that, when Jack Hubby and Richard Lewontin published this paper, and this is a classical sort of back-to-back. These are two back-to-back papers that Hubby and Lewontin published in '66, and this was a technique to study the presence of polymorphism in a population directly, right? And this their study was done with a particular species of Drosophila called Drosophila pseudoobscura. But the following years also saw people doing experiments with human proteins. And I think it's worthwhile just reading a couple of paragraphs from the paper of Hubby and Lewontin. So remember this. So I mean, in a sense, Dobzhansky was far ahead of his time because he was foreseeing these problems, which after 10 years, people were just coming up with. So he says that a cornerstone of the theory of evolution by gradual change, remember gradual change, this was the debate between Mendelians and biometricians. Biometricians saying that small changes get acted upon by selection, is that the rate of evolution is absolutely limited by the amount of genetic variation in the evolving population. And this is clear that if there is no genetic variation in the population, you can't have evolution. Fisher's fundamental theorem is a mathematical statement of this generalization. But even without mathematics, it is clear that genetic change caused by natural selection presupposes genetic differences already existing, on which natural selection can operate. So for natural selection to operate, you need genetic variation. In a sense, a description of the genetic variation in a population is the fundamental datum of evolutionary studies. Hence, we need a handle of what is the degree of genetic variation that exists in the nature, in a population. And it is necessary to explain the origin and maintenance of this variation and to predict its evolutionary consequences. It is not surprising then that a major effort of genetics in the last 50 years has been to characterize the amounts and kinds of genetic variation existing in a natural or a laboratory population of various organisms. And I think this just perfectly sums up the problem that existed there. The problem of quantifying variation, and that this variation is central to how evolution is going to take place, how natural selection will act. But we need to understand what is the extent of variation that's present in a population. What does Fisher's fundamental theorem say that he's talking about here? We discussed this in one of the classes. What does Fisher's fundamental theorem say? Anybody?
The rate of evolution depends on the variation present in the population.
Yeah, exactly. So the rate of evolution, by which we can say the rate at which fitness increases, is directly proportional to the variation in fitness that exists in a population. Fine. I mean, there are two terms to it, but let's not get into that. But perfect. Great. So there's one more paragraph that we should read here. So he says that the techniques of population genetics have not allowed us to ask directly the most elementary question about the genetic structure of a population. And that question is, at what proportion of his or her loci can we expect a diploid individual to be heterozygous? That's really the key. Put in another way, this is the question of how much genetic variation is there in any given population, right? And these two papers are sort of seminal for this technique, which sort of led the developments that are going to happen in the next 20 years or so. And they highlight that why this has not been done and what conditions need to be met for somebody to come up with a strategy that could help answer this question. They say that the reason for our present lack of knowledge about the amount of heterozygosity per locus in a population is that no technique has been available capable of giving a straightforward and unambiguous answer, even under ideal experimental conditions. Any technique that is to give the kind of clear information we need must satisfy the following criteria. 1. Phenotypic differences caused by allelic substitution at a single loci must be detectable in a single individual. Right? We should. The first criteria is that we should be able to separate out the two alleles that are present in a single individual. Allelic substitutions at one locus must be distinguishable from substitutions at other loci. Our technique should be such that we are looking at one particular locus and nothing else. If we suppose we take blood and we are characterizing the different proteins in blood, I should only be concerned about the two protein molecules that come from two alleles at the same locus, and all other proteins in blood should not matter for the analysis, for the technique that I am able to come up with. A substantial portion of allelic substitutions must be distinguishable from each other. This is important because what he's saying is that suppose that he's recognizing, or they are recognizing, that whatever this technique might be, no technique is going to be of the level of sensitivity that it will detect every possible allelic substitution in a protein. Suppose I have a protein and it's carrying leucine, and I pick up a mutation and that leucine becomes isoleucine. Now, it's possible that this change is so small in terms of the physical and chemical behavior of the protein molecule that my characterization technique does not allow me to capture that change. And they recognize that no technique is going to be perfect in terms of capturing all the allelic variation that exists. But whatever technique one comes up with, it should be able to capture most of this allelic variation that exists. All is not going to be possible. And finally, loci studied must be an unbiased sample of the genome with respect to physiological effects and degree of variation. This is really a detail. Essentially, they know at this point that human DNA contains thousands of loci, and hence, when you're making a generalized claim that in a population at most loci there is considerable heterozygosity, you are not doing it in a fashion that you sampled each of these thousands of loci. What you're going to be studying only a handful of loci and then make a generalized comment about all other loci. So how you select these handful of loci is important, and it should be unbiased. It should be an unbiased selection, such that whatever analysis you draw, whatever conclusions you draw from it, are extendable to all the other loci on the genome. All right. So, I mean, we can go through we will go through what their technique was, but can you think of something? Anything? How would we do this?
Maybe using some kind of, like, if there is homozygosity, like there should be phenotypically, like, drastically different features, characteristics.
Right. But
Like maybe we can tag like the.
But tagging is difficult, right? I mean, we don't know how to do tagging.
Okay.
Right?
Maybe we're mating and seeing the progeny then.
But then you don't know whether you're studying one locus or several loci. So why do molecules of DNA migrate on a gel?
Negative charges to positive.
Correct. I mean, they're negatively, DNA is negatively charged, and uh, when I pass DC current through it, DNA is going to migrate towards the positive terminal. Perfect. And suppose I were to do the same for proteins, what will happen?
Size wise.
Right. So for proteins, the separation is going to be dependent on the ratio of charge to size, right? If I run a gel like that for proteins, then suppose I take my blood and isolate all the proteins from it and run all these proteins on a gel, then these proteins are going to separate based on the ratio of their charge to size. And once I have this separation, you can imagine this gel on which I have separated all the proteins that are there, assuming that there is considerable difference between the charge to mass ratio of the of the different proteins that are present in blood. And then I'm going to have this gel on which all the proteins in my blood have separated. Now I still can't see them, but they're there somewhere. But I have these different bands, just as you have these bands in a DNA gel. You have these different bands, each of which is a separate protein molecule. If we get that far, what do you think we can do with that? And remember, we are saying that we will be thinking of only those proteins which are catalytic in nature. Any ideas?
So we can see how much the thickness of each band is, and
Well, so that's not going to be possible. Uh, but how will you see that band? You're not seeing that band, right? Does that make sense? So let me give you a sense of
We
this
can
gel.
compare. Okay.
Yeah, just 1 second. Uh, so the gel, Yash, you've not seen a DNA gel, have you?
no.
Yeah. So this gel is going to be a very jelly-like structure with a thickness of approximately this phone, right? And proteins are going to be migrating inside this gel matrix, and the weight of the proteins is essentially zero as compared to the weight of the gel, right? So the proteins are there somewhere in that gel. They have been separated according to their charge to mass ratio, but I don't know where they are. So the need for me is to find out where the protein of my interest is, right? And the help that I have is that I am looking for only that specific protein which carries a particular specific reaction in the cell. Does that make sense, Yash?
Yeah.
Yeah. Okay, Sangamitra, go ahead.
Oh, sir, it's fine. It doesn't make any sense anymore.
Okay. All right.
So in this gel itself, we can introduce the reactants of the reaction,
Uh-huh. And
and if the product is somewhat physically separable,
yeah,
say it shows some fluorescence or something,
yeah,
we can identify the band easily.
absolutely perfect. That's exactly what they did. Okay. Uh, great. So that's exactly what they did. So now we know the substrate, right? We know that the bands have separated. Let me just show you a couple of pictures from this. So, I mean, their methods is actually very well described. This is a extremely nice paper, and the methods are in pretty good detail. Let's take a look at this. This is the start of their gel. So this is where they loaded all the protein molecules, right? Don't worry about the dark bands here. Then they pass current through it, and this is the direction of migration. And these are the different individuals in the population. So they are studying a dozen individuals in the population. And what they do is, so I think these are esterases, but don't worry about the chemistry. Strain analysis for esterase. Yeah. So what they are doing is they are letting the gel run for a certain time, and all the protein molecules, all the different proteins in the sample of an individual have separated. So there'll be a band here, there'll be a band here, there'll be bands everywhere now. Now they are interested in that specific band which catalyzes the particular reaction which is catalyzed by this enzyme. And for that, they are adding in some cases the real substrate labeled, or in some cases another substrate which sort of which mimics the original substrate of the enzyme, such that you will get a stain on the gel wherever this reaction took place. And now after allowing this reaction to go on for a certain while, you're just imaging the gel to see for where all staining took place. So let's look at this individual one. What can you say about the homozygosity or heterozygosity of this individual? Anybody?
If there are multiple bands, then we can say it is heterozygous.
Yeah, so individual one is
Homozygous.
homozygous, right? Because there is only one band, so all the protein is present here. This individual, for instance, is a heterozygous individual. Well, so they comment in the paper that there appears to be a third band here, and they really don't know what this third band in the middle is. But essentially, they're saying that this allele is the same as this allele, which individual 3 is carrying. So individual 4 here is heterozygous, where one of the alleles is the one that's carried by individual 3, who is homozygous with allele with this particular allele. And the other allele in individual 4 is this allele which individual 5 is homozygous for, and so on and so forth. So single band indicates homozygosity and multiple bands indicate heterozygosity. So you can see there are some individuals which are heterozygous. So individuals 10 and 11 are heterozygous. Individual 4 is heterozygous. The others are all homozygous. But there is considerable allelic variation that exists in the population. So we have 1, 2, 3, 4. We have at least 4 different alleles which are present in a population of only a dozen individuals. Does the method make sense? Yes, no, don't know. Yeah, go ahead. Boitra, go ahead.
Okay, so even if it is heterozygous, there'll be only one allele expressed, right, because of dominance traits.
No, that's not how it's going to work out. So, no, so dominance doesn't work like that. Dominance doesn't mean, suppose I am carrying an allele small a and a capital A, and capital A is dominant over, suppose I'm carrying alleles A and B at a particular locus, and A is dominant over B. That doesn't mean that uh, the allele B was not being expressed in me. I am expressing both allele A and B. Dominance means that if you as an individual are carrying 2 copies of allele A, and I am carrying one copy of allele A and one copy of allele B, and you are expressing both the alleles, both the A's, which are identical, and I am expressing both A and B, if the physical manifestation that you and I have for that particular trait is identical, if you can't make out a difference between a AA and a AB, then we say that A is dominant over B. Does that make sense?
Yeah. Okay. Okay. Yeah.
Right. So an individual is expressing both A and B. Think of it like this: that how do I say this? Suppose for a particular reaction, you need, suppose for optimal physiology, you need 50 molecules of a substrate converted to product every second. Okay.
Okay.
Now you are carrying an allele, you are a AA individual, and each of these each of these A's can catalyze 55 molecules per second. Okay, so what is your total capacity?
110.
110. But how much do you need for optimal physiology?
50.
50, right? So you are operating at optimal. Does that make sense?
Yeah. Okay.
Right. Now I, as an individual, I am carrying one A and one B. The capacity of B is only 5 molecules per second. So what is my total capacity?
60.
60. But as far as fitness is concerned, do you think this will manifest as a difference between the two of us?
No.
Right?
Because you
And that
need
is,
only 50.
I need only 50, and that is what it means that capital A is dominant over B.
Okay. Right.
Okay.
Right. Yeah.
Now, if it was the other way around, that suppose for optimal physiology you didn't need 50, you needed 100, then you and I would be clearly distinguishable as far as fitness is concerned.
Yeah. Right.
Right? Because now I don't have the bandwidth to go up to 100. You can. So hence I would be lower fitness. And then we would say that A is not dominant over B.
Okay. Yeah.
Okay.
Right.
So that's how dominance works. But you
All right.
express both alleles. So in the example that we just saw, each individual is expressing both the alleles, and you can clearly make out, that whether where are these alleles. Any other questions, comments?
No, sir. No.
Anybody?
So there are two proteins because each strand will give out.
Yes.
Right. Okay.
Yes. Right. So I mean, suppose this esterase is present on chromosome 3, but I have two copies of chromosome 3, one that I've inherited from my mother, the other one from my father. Right. So I have two copies of this esterase genes, and it's excuse me, and it's very excuse me, and it's very likely that the esterase copies that my mother gave me and what my father gave me excuse me, the two copies that I got from my parents are different from each other, and hence I'm expressing two different variants of this esterase gene, and these two variants are likely to show up as different bands on a protein gel if somebody ran this protein gel, and hence you'll be able to discover heterozygosity at a particular locus. Is the method clear? Yes, no?
Yes, sir.
Yes, sir.
All right. Do you think this method underestimates or overestimates heterozygosity?
Underestimates.
Why? Underestimates is correct. Why?
Because there might be two distinct proteins but giving out the same result.
Clean that statement. Say that.
So two different enzymes might be catalyzing the same reaction and giving the same results.
But that would be a case, but that would mean that you have overestimated heterozygosity, because suppose a locus on chromosome 3 and a locus on chromosome 7 are both catalyzing the same reaction, then you're going to see two bands on the gel, even though you are homozygous for the locus at chromosome 3 and homozygous for locus on chromosome 4, right? So you are homozygous individual, but it will show up as heterozygosity because you have two different loci catalyzing the same reaction.
Okay, so means each protein will have a distinct band, no matter what.
Yeah,
Okay.
right.
Yeah.
So that's not it. That's not the reason. This method underrepresents heterozygosity. Why, Anirudh?
I think it depends on the resolution of the band.
Exactly. Yeah. So, I mean, think of it like this, that most of the alleles that are going to be present in a population are not going to have wholesale changes in the amino acid sequence that's going to be present in the protein. These are going to be relatively small amino acid changes that are going to be present. I mean, towards the at the end of the lecture today, I'll show you a graph. The amino acid changes are going to be relatively small. And how you are studying this is running this protein mix on a protein gel, and your hope is that those handful of amino acid changes in a protein are able to, one, resolve these two bands, right? That it changes migration on the gel such that you see this as two bands. And second, both the bands are functional, because in my case I am carrying an esterase A and an esterase B, and you can resolve them into two bands. But then if esterase B is useless, it doesn't catalyze the reaction, then you're not going to see the band, right? I'm only carrying one functional copy. So on the gel, I'm going to come across as a homozygous individual, which actually I'm not. I'm a heterozygous individual. So you need and on the other hand, I'm carrying A and B, both are functional, but the differences are so minor that in the crude technique that Hubby and Levantin have proposed, you just do not see the resolution of these two separate alleles. So I am going to see a single band because A and B cannot be resolved on a gel. And what again that means is that I'm going to come across as a homozygous individual, whereas I'm not. I'm a heterozygous individual. So this method, by some considerable degree, underestimates the degree of heterozygosity that's present in the population. I'm actually not sure, Yash, if there are several loci which will catalyze the same reaction. I think it's pretty rare that you have several loci encoding a certain enzyme which catalyze the same reaction. The fascinating thing about biological catalysts is not the fact that they can catalyze one precise reaction, but the fact that they can catalyze only that one precise reaction and nothing else. And this is unlike the inorganic catalysts that we've studied as chemical engineers. Inorganic catalysts are just good catalysts. They will catalyze a whole variety of reactions. Proteins are the sort of catalysts whose, I think, the wonder associated with them is that they will catalyze only that one reaction. So it might be a rarity that you have two proteins catalyzing the same reaction, but not generally, right? I mean, I say that with an asterisk. There is a whole class of proteins which are called moonlighting proteins. And the main behavior of these moonlighting proteins is that in addition to the one reaction they catalyze, they also have an alternate function in the cell that they will catalyze some other reaction also. So there is specificity to a large extent that a protein will only catalyze one reaction, but there are some exceptions to that as well. All right, cool. So Habby and Lewontin published these papers in the 60s, and they note that actually there is quite a bit of polymorphism that exists in the population. So it looks as though it's not classical hypothesis, it's actually balance hypothesis that seems to be how evolution is operating in a population. All right. How many of you have heard of this term called molecular clock? Okay, Sakshi, who else? All right, Sakshi, tell us, what have you heard? Sangamitra. Okay, two of you, tell us what have you heard? What do you know of this?
Circadian rhythm, kind of. Like, there are different molecular clocks. Like, one example is a circadian rhythm, like that. And
okay. Okay. Oh, anything else? Yeah, Anirudh.
It comes in when we try to predict the neighboring an evolutionary tree.
Mm-hmm.
So at that point, the mutations are somewhat considered to be molecular clocks. The degree or variation,
Yeah.
so that would be considered.
Yeah, exactly.
Rate of mutation.
Sakshi, louder, I couldn't hear you.
Yeah, something to do with the mutation rate, maybe.
Okay, sure. So so Sangamitra, the molecular clock aspect is not used in the context of a circadian rhythm, which obviously is also a clock. But the molecular clock is something that was proposed in a series of papers that were written by Linus Pauling and his postdoc Emil Zuckerkandl. And in the mid 50s, they proposed this idea, what is called the molecular clock. And I'll just discuss some nuances of that, and then we'll pick this up on Wednesday. So hemoglobin actually is a protein that is very, very well studied from ages, right? So you have these scenarios where some model systems, be it a particular protein, be it a particular organism, were chosen historically to study a certain phenomena. And because these were the early choices of people, and knowing about a system is pretty hard. So if you want to address a certain question, you want to work with a system where a number of other facets of that system are already known. So you have a foundation to build, or you have a foundation to design your study on. And hemoglobin is one such historical accident, that early researchers were very interested in hemoglobin. And hence you knew a lot about hemoglobin. And then subsequently, whenever you were looking at a protein candidate to study one question or the other, hemoglobin was an obvious choice because a lot of its biochemistry, genetics, and sequence information was already available. So hemoglobin forms a large part of classical genetics and evolution textbooks. And this plays out at the level of organisms also. I mean, Saccharomyces cerevisiae as a yeast, as a model organism for studying eukaryotic genetics, is big, but it's actually a very, very finicky organism. It's not easy to work with at all. But because shifting to another organism now is inconceivable because of the amount we know for cerevisiae, we are stuck with these historical accidents. All right. So hemoglobin, around 50, Pauling and colleagues, Zuckerkandl had not joined then, showed that sickle cell hemoglobin had significantly different electrophoretic mobility compared to normal hemoglobin. Right? So again, these are two different alleles showing different migration patterns on a protein gel. Remember, this is many years before Hubby and Lewontin published their paper. But Pauling and his colleagues did not place this particular experimental technique in the context that Hubby and Lewontin did. So Pauling was the first to think of evolution at a molecular level. Ernst Mayr, who we will revisit in the last part of the course when we are discussing speciation, was always interested in evolution at a morphological level, and he thought that was the correct way of looking at evolution. But Pauling was actually the first one who was comparing sequences. Going back to hemoglobin, there was a book that was published in 1909 that actually showed that the shapes and angles of the crystals that you get from hemoglobin from different species are actually different. So Zuckerkandl, who sort of, I think he was in Austria, and World War II started, and his family fled to Algeria. I mean, I think it's actually a pretty sad story. Eventually, he met Pauling in Paris in '59. He had applied to Pauling, and I think Pauling was interested in questions of chemistry that Zuckerkandl was proposing. And eventually, he joins Pauling at Caltech in '59. And their initial idea to working on protein chemistry is discarded, and Pauling asks him to work on hemoglobin. So, and in the following year, these three guys publish a paper on hemoglobin, and they note that the gorilla, chimpanzee, and human patterns are almost identical in appearance. And as one gets further away from primates, the amount of primary structure that is shared with human hemoglobin decreases. So they are proposing some sort of a relationship between a protein structure along with the evolutionary distances between two different species. So at that time, sequences were becoming available, and protein sequences, and Zuckerkandl, he got access to the alpha and beta hemoglobin chains, and he found that these two were extremely similar to each other in terms of amino acid sequence. And this sort of led him to think of that this was a duplication accident that had taken place. So, well, a long story short, I think we don't need to worry about these details. But for any particular species, this is the key here, that for any, if we take a pair of species, let's say human beings and horse, we know from fossil evidence the amount of time that has gone since human beings and horse separated from the common ancestor that human beings and horse shared. If we plot that distance between any two species, so this x-axis is the time since the common ancestor of any two species that we are looking at. So dog and cat could be one, horse and cow could be another one, anything. So x-axis is time since common ancestor for two species. Y-axis is the amino acid differences. If I'm looking at, so hemoglobin is pretty well conserved. So if I'm looking at hemoglobin sequences in these two species, and I'm actually counting the number of amino acid differences that exist between these two species, and that forms the y-axis. So the x-axis is how much time has gone since these two diverged, and y-axis is that for a particular gene, how many amino acid differences exist for these two species. And if I plot these two, so each point represents the data for a pair of species. And if I plot them, I get a roughly straight line. And this is the idea of molecular clock that Pauling and Zuckerkandl proposed in these series of papers that they published in the 1950s. That it's almost as if there is a clock ticking, that every 10 million years or so, one amino acid change takes place, right? So there is this clockwise phenomena to how evolution is proceeding in proteins at a molecular level. Now, people like Ernst Mayr were not really concerned about that. They said this has nothing to do with evolution. Evolution is more at a more morphological level. But to Pauling and Zuckerkandl, this was extremely interesting and had big implications. But then it also poses a question that what sort of underlying mechanism can explain this clock-like behavior of amino acid changes between these two species? Let's just take a look at a few other proteins. So this is,
Sir,
yeah, go ahead.
my question is, up until now, all the studies were about sudden changes or non-periodic changes which are occurring. So why did these
Which
guys
studies are you
go
referring
after
to?
any, like we study mutations in evolutionary studies, right? In general, up until now
Right,
we had many examples about mutations taking place or
right,
environmental factors changing.
right.
So why did these guys study about periodic changes occurring?
No, they're not studying periodic changes. The idea is that, so in the premise to the article that Pauling and Zuckerkandl wrote, they say that there are three types of molecules. Okay? The first type of molecules are called biological polymers, and these include, they say, these include DNA, RNA, and protein molecules. The second type of molecules, they say, are the compounds that that enzymes, protein molecules, act on. So substrate A going to product P is the second set of molecules. And the third set of molecules are molecules that are present in the environment that an individual is interacting with. So glucose, if it's present in the environment, an individual takes it up for consumption, is the third set of molecules. And they say to understand the evolutionary behavior or the evolutionary history of an organism, the most information that's contained is in the first set of molecules, which is DNA, RNA, and proteins. They give each of these classes of molecules a name, which I don't remember. They're just, I think, just obnoxious names that they give. So they say they are studying, their premise for studying these molecules is from an almost like an information theoretic point of view, where they're saying that these molecules and their sequences contain information regarding the historical past, the evolutionary historical past of these species, and hence we are studying and comparing them. That's one, right? So that's the premise of for them studying. DNA sequencing is not yet available. Protein sequences is, and hence they are focusing on proteins. Now, they conceive that there is a evolutionary commonality as far as origins of species is concerned, and hence, and there is information regarding when divergence of different species took place. So they are looking at different species and counting the number of differences that exist in the amino acid sequence of a particular protein. And what they end up with is this very curious relationship that the trend between how many amino acid differences and how far the two species, how long back the two species diverged from a common ancestor is a linear one, right? And if it's a linear trend, then it means that if I and another species diverged 10 million years ago, so the rate is about, for hemoglobin, the rate is about one amino acid change per 11 million, but we can take it 10 million. So the rate for hemoglobin is about one amino acid change in 10 million years. So if I and another species separated 10 million years ago, then we have one amino acid chain. If I and another species separated, had a common ancestor 20 million years ago, we have two amino acid changes. If I and another species had separated 30 million years ago, then we have three amino acid changes if we were to compare the sequences between the two of us. Now that is just an observation that they have, right? There is no answer for that as yet. But that is an extremely interesting observation, and they call it molecular clock, that whatever be the underlying reason for this behavior, what the manifestation of it is, that there is almost like a clockwork feature in which evolution is working. Why should it be that every 10 million years you should have one additional amino acid difference between two different species, irrespective of what those pairs of species were? That seems incredible. So the clockwork phenomena comes from there. Your other point, I didn't really understand that. It's too drastic. I didn't really understand that other part.
What I meant was, in general, evolutionary studies are about changes happening
Yes.
in the DNA sequence
Yes.
whenever DNA was discovered after that.
Yes.
So those changes were generally in lab experiments, stimulated by mutating or
Yes.
radiating them or
Yes.
changing the environment and so and so. But
Yes.
these studies which they Pauling and
Yeah. Zucker
Zuckerkandl were
candle.
doing,
Yeah.
these were like after the observations they seemed periodic. So I thought they were having some prior knowledge about this periodic change.
No, no, no. There is no one has any prior knowledge of periodic change, right?
Okay, okay.
And evolution experiment, while the first evolution experiment was done with bacteria, was done in the 1880s, nobody is doing evolution experiments in lab up until this point. I mean, evolution experiments in lab are only a product of the last 20 years or so. Uh, so there is none of that going on. What they have come across is just this astounding discovery that sequences of different species seem to be following. That's all we have so far, right? And once we complete this section of what this will eventually lead to is a famous theory that was proposed by first in 1968, but then exhaustively expanded in a book that came out in 1983. Uh, once we finish this once we finish a discussion on that, we will come back to the next section of the course is just dedicated to experimental evolution, and then we will. I think I will be better able to answer and understand your question. But
Okay.
as far as now is concerned, we have this very curious observation that protein sequences are diverging in a clock-like fashion. And let's take a look at, so these three are again, these three proteins are again looked at from, this data was available at that time also. So after hemoglobin, the two other proteins that were studied were cytochrome C. Don't worry about functions, okay? Cytochrome C and a peptide called fibrinopeptide. And the slope of the straight line would change depending on which protein you were looking, but the molecular divergence between species for a particular protein seemed to follow this straight line relationship. So it's almost as if that I know the slope of hemoglobin line, and now I have two species for which, for the pair of which I do not know when the common ancestor was, right? I have no knowledge of their evolutionary history. What I can do, I can just look at their, I have this reference line for hemoglobin from all the other species that I've looked at as pairs. So I can look at these two unknown species, which I can't trace back to a common ancestor in the past, and I can just sequence their hemoglobin uh, protein sequence, amino acid sequence, and I can just say that, okay, this is 5 differences, and I know differences accumulate at 1 every 10 million years, and hence their common ancestor lived 50 million years ago. Right? So that's the power of the molecular clock. But my question to you is, how can we explain the phenomena of molecular clock in the prism of what we know so far? Any idea? Is the question clear? Why this linearity? Why should it be linear? And in the theoretical construct of evolution that we have, how can we justify this linearity? Any ideas?
Linearity as in like uh, it's just the variation is increasing, like it's a continuous process. Maybe that's how we can justify.
No, but why linear? Why does it have to be clockwise? So the idea of molecular clock is that the number of differences is directly proportional to the time since these two species diverged. If two species diverged 100 million years ago, the number of differences is 10 times than the species that diverged 10 million years ago. Why should evolution follow this clock-like fashion is the question. Does that make sense, Anita?
Yes, sir.
That's the question. I mean, nothing in evolution that we have studied so far would suggest that this kind of a linearity should exist. Beneficial mutations can occur all the time, and as species are evolving their sequences, maybe all the beneficial mutations get saturated, and then there are no more mutations to be picked up. And the slope of the straight line is not constant. Some proteins are diverging very fast, like fibrinopectins. But some proteins, like cytochrome, actually do not pick very many mutations at all. The slope of that straight line is very small, right? So, I mean, just to emphasize that, cytochrome is diverging very slowly, right? Amino acid change, the y-axis is amino acid changes per 100 amino acids. So you have 20 amino acid changes. So you have 20% amino acid changes taking place in 400 million years, right? So in 400 million years, you have roughly 20% protein sequence change. However, in 400 million years, you have the entire protein change for fibrinopectins. Fibrinopectins, if the species are more than 200 million years further apart, then there is no sequence similarity between the two species. So different proteins are diverging away from each other at different rates, but for a particular protein, there is linearity, right? And I will start the discussion on Wednesday by seeking explanations from you that what have we studied so far which would explain this linearity. In what context should I use the theoretical understanding that we have gained so far that can explain this clock-like work in which evolution seems to be operating at a molecular level? And then we will discuss this extremely famous paper by King and Jukes. This came in Nature in 1969, and it's called non-Darwinian evolution. And this was an explanation. This was preceded by a paper in 1968 by somebody called Moto Kimura, which is just a 2-page paper. But Kimura's paper is essentially math. But this paper, which came the following year, is a much nicer version of Kimura's argument, and it's more readable. So we'll discuss this paper, and this sort of helps explain that why we should be witnessing this linearity in nature. And we'll start from there. Okay. So if there are no more questions, we can stop here. And remember, the assignments are due. The midsem assignments are due on Wednesday before class. So if there are a couple of short questions, I can take them. Otherwise, I will see you guys on Wednesday.
I had sent you a mail regarding a few.
I will answer that. Yes.
Okay.
I'll get to it today. Okay. All right. Thanks, everybody. I will see you guys on Wednesday.
Thank you, sir.
Thank you,
Bye.
sir.
Bye.
Bye, sir.